and welcome in this new appointment with the Redox webinars. Today we open this new session, which is dedicated to the oxidative stress as a therapeutic target, from neurodegeneration to sickle cell disease, and we have three very eminent and authoritative speakers, which are Professor Joao Laranjinha, Professor Mutai Aslan, and Professor Rita Maccarone, which are going to put a very good contribution to this very important and interesting topic. So we have three talks, and for any question, I strongly invite the audience to post them on the chat, and I'll submit them to the speakers by the end of the three talks. So the first one is Professor Laranjinha. Professor Joao Laranjinha is Professor of Biochemistry and Molecular Biology at the Faculty of Pharmacy at the University of Coimbra in Portugal. His research is mostly focused on the functional impact of nitric oxide in the brain as a neurometabolic regulator and as a mediator of neurovascular coupling with impact in aging and neurodegeneration. He has overall published over 100 papers in many prestigious journals, and he has coordinated over 20 external funded projects. And today he's going to talk about redox regulation of neurovascular coupling by nitric oxide to improve cognition. Please, Joao, the floor is yours.
Okay, let me share the...
Okay, it's working.
Okay, so good afternoon, everybody. It's a great pleasure to participate in this webinar series, and I'd like to thank Professors Mauro Maccarone and also Alexandro Liuti for this kind invitation. So I'm Joao Laranjinha from the University of Coimbra, Portugal, and I'll be talking about the title is here, the redox regulation of neurovascular coupling by nitric oxide in order to improve cognition, particularly in aging and neurodegeneration. And oops. Okay, so this is the plan for my talk. I will briefly address some key concepts on neurometabolism, and then the neurovascular coupling concept. So the brain is equipped with fine mechanisms to regulate cerebral blood flow, CBF, as a function of neuronal activity in a precise temporal and spatial way. So I will address this concept very briefly, and then we'll show some of our data supporting our contribution actually to elucidate the molecular mechanisms underlying this neurovascular coupling concept, which is critical for brain function and structure. I will briefly address in this regard some of the peculiar physical-chemical properties of NO, nitric oxide, as a cellular messenger, and also the technology that we have used to assess our hypothesis. Then I will move to a second part, trying to illustrate that decreasing nitric oxide bioavailability is linked to, in a causal way, to cognitive decline. Maybe I will have to skip this part because I was prepared to speak during 45 minutes, but actually I have only 30 minutes because I want to go to this new part here, the last part, which is in a way it's a molecular solution to this problem of impairment of neurovascular coupling. So can we devise strategies, molecular strategies, to maintain nitric oxide bioavailability, neurovascular coupling, and in this way to improve cognition? So this is a new part, not published yet. So I think maybe it's the most interesting part. Okay, so just one slide on these key tenets in neurometabolism. I mean, everybody knows this, but let me just summarize that we mammals conversely to other vertebrate species, we use about 20% of oxygen and glucose in our brain, and in our brain represents only 2% of body weight. So this is a heavy energy budget in our body, the brain. On the other hand, the brain has little capacity to store glucose. There are some glycogen in astrocytes, but not that much. And there is also a protein, neuroglobulin, in neurons, but nothing compares with the myoglobin concentration in muscle. And also, very importantly, the metabolic activity of the brain is constant over time, but there are fluctuations. There are, in this baseline, there is an increase, a transitory increase in cerebral blood flow in connection with localized neuronal activity. So the normal brain function requires this tight regulation in terms of time and space of the cerebral blood flow as a function of neuronal activity, because there is this close association between the two phenomena. The neuronal activity, mostly driven by glutamate, and the CBF. And this is known from this has been known since the 19th century from the work of Roy Sherrington. There is a beautiful paper in J Physiology in the 19th century describing these mechanisms. So for the brain, this is a nightmare, of course, to allocate energy, resources among regions according to neuronal activity. So this is the concept of neurovascular coupling. The functional connection between neuronal activity and increase in cerebral blood flow, also called the functional hyperemia. And the paradigm for this connection, this functional connection, this fine equilibrium between energy demands imposed by neuronal activation and substrate delivery, glucose and oxygen, by the vasculature. The paradigm for this is so-called astrocytic bridge that I'm going to describe very briefly. So this process, this communication that is tightly regulated, as I said, in terms of time and space, is largely driven by glutamate. So upon glutamate release in synapse, interaction with metabotropic glutamate receptors in astrocytes, this can activate the increase of calcium. The subsequent increase of calcium can activate some target enzymes like phospholipase A2. So the resulting arachidonic acid can be the substrate for several enzymes localized in astrocytes or in the endothelial cells, giving rise to several vasoactive compounds like prostaglandins, like epoxy derivatives, which dilate and increase the cerebral blood flow, or some hydroxylases, which give rise to substances that constrict, decrease the lumen of this artery. So this is the paradigm, the astrocytic bridge. There are other players in this scenario, and one of the players that I'm focusing in is this enzyme here, the neuronal isoform of nitric oxide synthase. So located in the postsynaptic neurons, there is the glutamate NMDA receptor, and so the subsequent increase of calcium can activate this enzyme, producing NO that by itself directly can interact via the canonic way in the smooth muscle cells, in the vessels, with the soluble guanylate cyclase, and so the decrease in calcium will induce the vasodilation, the increase the cerebral flow, and this area here will have more glucose and oxygen. By the way, this connection is so tight between neuronal activation and the increase in CBF that the CBF has been used in the magnetic resonance studies as a proxy for neuronal activation. So what we see in these nice pictures of functional magnetic resonance imaging is not really neuronal activation, is the release of oxygen from hemoglobin. But before describing some of these results that led us to this hypothesis of nitric oxide directly, the direct mediator in this process, let me just focus a little bit on the peculiar properties, physical-chemical properties of nitric oxide. And it is important to recognize that nitric oxide is an intercellular messenger because it diffuses faster than it reacts. Conversely to what is often written in many papers, nitric oxide is not particularly reactive. Actually, it's pretty stable in the absence, at least under normal physiology, is pretty stable, is as stable as oxygen. It's hydrophobic, so it's freely diffusible, it permeates very easily biomembranes, and diffuses very, very fast. In fact, some time ago in these publications here, we have calculated the half-life of nitric oxide particularly in an hippocampus, okay? In the brain, in rat hippocampus, it's about, the half-life is about half a second, and diffusional spread of nitric oxide under the conditions where we perform our experiments is about 200 microns. Just to give you an idea, the diameter of a neuronal body is about 10 microns. The diameter of a red blood cell is 10 microns. So the diffusional spread of nitric oxide in the brain in hippocampus of rats is 200 microns, okay? So this led us to conclude that a single molecule of NO can move out and in between cells many times with this time span. And these notions permit to understand this concept of volume signaling. That is, this is a picture of hippocampus in the screen. Not all cells synthesize nitric oxide, but when a group of cells simultaneously produce nitric oxide, the concentration of nitric oxide within a single cell is much greater than if that cell was producing nitric oxide alone. So this is the concept of nitric oxide, and we can imagine this volume signaling as this diffusional spread of a kind of wireless connection between cells. So nitric oxide can integrate the activity of different neuronal cells, regardless of whether they are connected physically by synapses. So this is an important notion to understand the role of nitric oxide as a messenger in neurovascular coupling. So the idea is that nitric oxide conveys information associated to its concentration dynamics, the volume signal. And so this poses a problem. How are we going to measure this? And to quantitatively address this issue, we had to measure the concentration dynamics. I mean, the profile of change in time and space in the brain, in vivo, of course. We have developed special tools along the years with my dear colleague Rui Barbosa. We have developed micro sensors and micro biosensors. Micro means some of the sensors based in carbon fibers with the 7 microns diameter. So the diameter of these fibers are smaller than the diameter of neuronal body. And also in a collaboration with the Center for Microelectrode Technology, University of Kentucky, with Greg Gerard, we have developed a ceramic-based multi-site platinum microelectrodes. And now we have developed along the years several microelectrodes, microbiosensors to measure simultaneously in vivo not only nitric oxide, but also oxygen, ascorbate, glutamate, glucose, lactate, etc. And we can mount all these sensors in arrays consisting, for instance, with microinjection pipettes, with laser Doppler flow probes, commercial laser Doppler flow probes to measure cerebral blood flow. And these arrays have a predefined geometry, so we can do some calculations regarding the diffusion, and we can insert these arrays stereotexically in the region of interest. Regarding the models, we can use these arrays in acute brain slices, in anesthetized animals, and also in vivo in freely moving animals. So we do the surgery, we implant the arrays, and then after recovering a couple of days, we connect the arrays to the potentiostat, and so we can relate the behavior with the dynamics of the neurometabolites, including NO and oxygen, in real time. So this is a real-time connection between behavior and the dynamics of these neurometabolites in the brain. So I'm going to show you, I'm going just to illustrate or to show you some data, not exhaustively, of course, but some important pieces of data that support the role of nitric oxide produced by the neuronal isoform, not by the endothelial isoform, as the direct mediator of neurovascular coupling. So this was measured simultaneously, the blood flow and nitric oxide using our arrays in the dentate gyros of the right hippocampus upon glutamate stimulation. So in blue, you will see nitric oxide, and in red, blood flow. And so upon stimulation with glutamate, we have a transient increase of nitric oxide from a background that is coupled in time with a transient increase in cerebral blood flow. And so we can repeat this several times. If we look carefully, the transient increase in nitric oxide from a background comes a few seconds before the transient increase in blood flow, as we would expect if nitric oxide was the diffusible messenger between neurons and blood flow. Of course, we have done a lot of pharmacology. If we inhibit the source of nitric oxide, the enzyme, we decrease both blood flow and nitric oxide. If we inhibit the NMDA receptor glutamate the same way. If we inhibit the endothelial isoform, nothing happens. So this nitric oxide is coming from this isoform, not from this isoform. And of course, considering the canonic pathway for nitric oxide activity, if we inhibit the soluble guanylate cyclase, we get the NO signal, but not cerebral blood flow. So, by the way, these transient increases in blood flow occur in the absence of any significant modification of the mean arterial blood pressure. So this is a very localized event. In the localized, I mean, as I said, the diffusional spread of nitric oxide is 200 microns. So this event occurred in a sphere of tissue of about 200, 300, 400 microns. Okay, in the absence of any change in mean arterial blood pressure. So overall, these in vivo dynamics accomplish the fundamental criteria for the neurovascular coupling concept, which is a temporal, amplitude, and spatial relationship. What is the prediction? If we increase the cerebral blood flow following the transient increase of nitric oxide, we would be able to measure or to observe a transient increase in oxygen, and now registered here in green, and that was the case. So we have the nitric oxide, we have then in red, this is real time, okay, and simultaneously, in vivo, in the red brain, in the hippocampus. And then in green, we have the transient increase in oxygen. And of course, there is a nice relationship between the amplitude of the blood flow and the oxygen. Also, just to the final piece of data to convince you that really nitric oxide coming from the neuronal isoform is the diffusional messenger. in this communication between neurons and vessels, mainly arterioles in this case, is that a key tenet for neurovascular coupling concept is that the cerebral blood flow, the CBF changes, should be graded according to synaptic activation, according to neuronal activity. And that is what is shown here. So we have a nice relationship between the amplitude of the NO peaks and the amplitude of the cerebral blood flow changes. So, I mean, I think we can conclude that even if our understanding of neurovascular coupling is incomplete, there are other pathways, other mediators. The nitric oxide, the neuronal nitric oxide derived from this enzyme here is a key link in this mechanism supporting neurovascular coupling and functional hyperemia. Most of this was published already. This was the first paper, the first fundamental paper here. So, in addition to the, let's say, the astrocytic bridge, there is this, let's say, less complex mechanism based on diffusion, which is the wireless connection between neurons and vessels via the diffusion of nitric oxide coming from this enzyme here. Well, this must sound like an awkward idea, very eccentric idea. I mean, if neurovascular coupling is a process that is critical for brain structure and function and for cognition. So the brain relies in these non-canonical pathways via volume signaling that is characterized by the absence of recognition by receptors, as the traditional neurotransmitters do. This sounds a little bit strange, but if we think carefully about this, it is not. In spite of being this communication is being supported by diffusion via volume signaling, still remains a highly intrinsically controlled mechanism. Why? Because of this, and this is published in these papers there. Upon neuronal activation, we've got NO production, NO diffusion to the vessels, and increasing blood flow. And we have shown that in the brain, the main mechanism for nitric oxide inactivation is the reaction with oxyhemoglobin. So by increasing blood flow, NO switches on its own mechanism of inactivation. So we have a switch on and we have a switch off mechanism. So this is intrinsically a highly regulated mechanism. Okay, so the second part, let me say, okay, I have more 10 minutes or so, right?
Yeah, absolutely.
Okay. So in this second part, I was going to illustrate some this correlation between the impairment of neurovascular coupling, NO bioavailability, and cognitive decline. But maybe I have to skip this to move further to this new molecular mechanism. But let me just say the following. Let me put it this way. When the microvessels of the brain do not work, cognition does not work either. Okay? So the idea is that vascular health is fundamental to cognitive performance. Let me just show you this nice study from 2008, but this is a nice study with two populations. One population of high risk of developing Alzheimer's disease. It doesn't matter, what is high risk here, but just for the sake of the time, it's family history and apogeny E4 gene, and the low risk. So the high risk population are the reds, and the low risk are the blues, and they are submitted to a typical task of hippocampus, shown here, which is the face name encoding test. So you have a name, you have a face, and when you are establishing this relationship between the name and the face, the bolt signal and the blood flow is registered, and you can clearly see here that the blues, the low risk, have increased blood flow and have stronger bolt signal as compared with the high risk. So this is in humans, and these and many other studies, just to be brief, permit to conclude that there is a correlation between CBF changes, CBF dysregulation, and cognitive decline. This has been established in preclinical models in rodents, in others, etc. The problems here and the major questions are what is the neurobiological link, of course, and if there is causality between the CBF changes and the decline. And in this regard, one must recognize that the functional connection between neuronal activity and cerebral blood flow changes, I mean neurovascular coupling process, implies that an impairment of this process leads to a progressive decline in cerebral blood flow, increased neuronal dysfunction, and cognitive decline. So all along this hypothesis here, this is from Zlokovic, which is one of the main authors in this field. So, under mild hyperfusion, there must be a compromised protein synthesis. So the synaptic plasticity is compromised, supporting learning and memory. And for more severe hypoxia, we can have a strong decrease in ATP synthesis, so the restoration and ionic gradients are not operating anymore, decrease in sodium potassium TPA, so the neurons are unable to fire action potentials, and we go all the way to the in this cascade. So the message is there from the normal to the Alzheimer's, or Alzheimer's is the major risk for dementia associated with the vascular dementia, are account for almost 100% of cases of dementia. So the compromise, the impairment in neurovascular coupling is not only a component of the vicious cycle that is established between the decrease of oxygen and the cell death, and supporting all these toxic cascade from age-related memory impairment, MCI, synaptic failure, and cognitive dysfunction, but is likely a early causative event of this cascade. That is a widely accepted notion nowadays. So we wanted to check if this NO-mediated, neuronal isoform, mediated neurovascular coupling could be an early impairment, an early event in this toxic cascade, disturbing the function of the mammalian brain as a kind of a black hole. And for that, we have used several models, rat models of Alzheimer's, the triple transgenic model, of aging, of diabetes, and we use several models because if we are able to make common observation across the several models, likely the observation that we are making is not specific of the model, but is of general occurrence. Okay, so that is why it's important to use several models. So I had here several, to illustrate just that before cognition impairment, before, for instance, A-beta deposits in Alzheimer, in adult rats, the earliest event in all these models before cognitive impairment is the dysfunctional of the NO-mediated neurovascular coupling. Okay, but I'm going to skip these slices here. So the main point here is the decrease in NO bioavailability in all those models is the early event that later leads to the animals to develop cognition impairment. Okay, there are two fundamental ways for a decreased bioavailability of nitric oxide. One is a dysfunctional or an impaired or not a no active neuronal isoform synthase activity. The other one is the reaction of NO with superoxide radical. As you know, the reaction of superoxide radical with nitric oxide is the fastest non-catalyzed reaction in biology. Actually, the rate constant of the reaction of superoxide radical with NO, the second order rate constant is 10 to the 10th molar minus 1 second minus 1, is higher than the rate constant for the dismutation of superoxide radical by superoxide dismutase. This means that NO competes favorably with SOD for the reaction with superoxide radical. Okay, and of course, if we produce in this reaction the superoxide radical, we don't know, we produce peroxynitrite. Is not a radical, but is a wonderful nitrating and oxidating species leading to oxidation, nitrating, whereas hydrogen peroxide is involved in signaling. So these are the two possibilities for the decreased bioavailability. Okay, I think I'm almost running out of time, but let me just show you the last part.
You can take a couple of minutes more. No problem.
Okay, thank you. Okay, thank you. I am sorry for this, but I was really prepared for 45 minutes, but no problem.
Absolutely no problem.
I think that the message is clear. Okay, is there a solution for this decrease in nitric oxide bioavailability? And so this is our hypothesis, the redox cycle of nitrite and ascorbate. Why? Okay, everybody knows this classical pathway for nitric oxide synthesis. So nitric oxide synthase is a highly regulated enzyme, several substrates regulated at several levels, protein-protein interactions, post-translational modifications, phosphorylase, etc., etc. Okay, this is a highly regulated process. L-arginine as the substrate. As you know, the nitrogen atom comes from the guanine moiety arginine, and the oxygen atom in nitric oxide comes from molecular oxygen. Okay, so molecular oxygen is a substrate for enzyme. That means, remember that line under hypoxia, the nitric oxide synthase is not operative because substrate is lacking oxygen. And of course, by this way, upon interaction with metal centers, hemes, and thiols, this is the paved way for nitric oxide regulation of biological process. The half-life of nitric oxide is largely controlled under these circumstances by its oxidation by 1 and 2 electrons. 1 electron oxidation to nitrite, NO2 minus, and 2 electron oxidation to nitrate, NO3 minus, okay, mainly upon reaction with oxygen, and in this case here with the oxyhemoglobin. But soon after the identification in 89, something like that, of the nitric of nitric oxide with the endothelial-derived relaxing factor, it was realized that if from here to here is 1 electron oxidation, we all know that 1 electron in biology is a piece of cake. So, I mean, it's very easy to get 1 electron to reduce back nitrite to nitric oxide. Ascorbate, electron transfer chain, and also polyphenols, and also under hypoxia and acidosis, and also the so-called non-dedicated nitrite reductases, as called by 2 colleagues of mine and friends, Luisa May and José Mora, molybdenum enzyme, xanthinoxyl reductase, aldehyde oxidase, sulfite oxidase, and also deoxyhemoglobin, deoxymyoglobin, cytochrome C oxidase, etc. So it's very easy to reduce back. So this is not an end stable product of nitric oxide metabolism, as often appears in papers, okay? So it can be a a biological precursor of nitric oxide. And also, very, very interestingly, we consume a lot of nitrate in the diet, green leafy vegetables, for instance, lettuce, for instance, beetroot. And also in our tongue, there is a community of bacteria that are equipped with a nitrate reductase. Okay, so these nitrate reductases can reduce nitrate to nitrite and also to NO, closing the cycle and supporting this new pathway, in addition to classic, well-regulated, oxidative pathway for nitric oxide synthase via nitric oxide production via nitric oxide synthase. We have this novel pathway, the nitrate, nitrite, NO pathway. And if you are interested in this topic, I just put this slide here from a good friend and collaborator, Jon Landberg from Karolinska, one of the pioneers in this area. So to show you that we keep with a recycling mechanism, an interocellular recycling mechanism for nitrate. So we ingest nitrate, part of nitrate is reduced to nitrite by bacteria in the oral cavity, and nitrate and nitrite are absorbed, and 25% of nitrate is secreting back in the salivary gland, so closing the cycle. We have done also a lot of work with what happens to nitrate and nitric oxide in the stomach, but it's not the point of this communication. Just to illustrate that, as I said a few minutes ago, nitric oxide synthase is a homodimer. This enzyme uses oxygen as a substrate. So, and this was realized by Julian Miller, that I met a long time ago, that, although nitric oxide is a vasodilator in cerebral blood vessels, the synthesis of NO from arginine requires oxygen. So this enzyme is not suitable as a mediator of hypoxia-induced cerebral blood flow vasodilation. So under hypoxia, this enzyme is not working properly. So our hypothesis is this recycling is applying nitrate, nitrite, and NO pathway to maintaining the neurovascular coupling in the brain and supporting the cognitive enhancement. We have done a lot of molecular mechanistic studies, preclinical models, and we are currently running a clinical trial with patients from the neurological department here from the University Hospital. Let me likely this is not going to be a highway, okay, more a highway like in the Led Zeppelin song, Highway to Heaven. It's more like a Beatles song, the long and winding road to demonstrate this hypothesis. But anyway, this is just to show you that, for instance, ascorbate is a wonderful reductant of nitrite to NO. So you can easily, if you mix nitrite and ascorbate, you get the doublet radical of ascorbate radical, and also with polyphenols like caffeic acid, you have the septate signal of the semiquinone radical caffeic. If you decrease the pH, you have increased nitric oxide. And this is going to be a very complex slide, but I think in 30 seconds I can describe it to you. A critical observation is that when we stimulate the NMDA receptor, glutamate NMDA receptor in the brain, we have observed the release of ascorbate from neurons. Neurons concentrate, the concentration of ascorbate in our blood is about 50 microns. The concentration of ascorbate in neurons is above 5 millimolar. What is doing this ascorbate in neurons? Of course, we can speculate antioxidant activity and so on, but I mean, 5 millimolars is really a lot. What we have observed is that upon glutamate stimulation of NMDA receptor, we have ascorbate in the extracellular medium, a lot of ascorbate, up to 500 micromolar. And we know that because we were able to develop the special nanotube modified microelectrode for ascorbate. And when we perform the glutamate stimulation, as we have done before, and measure the cerebral blood flow, And if we put a little bit of nitrite in the stimulation pipette, we get more NO, and we got an increase in circular blood flow. Okay, from here to there and from here to there. In another model, hypercapnia model, we observe the same in both models. Here there is no glutamate stimulation. It's just the decrease in oxygen, a kind of hypoxia model. And we have, in the presence of nitrite, we have more NO and more circular flow. So just to be brief, these data largely support the occurrence of this redox interaction between ascorbate released from neurons with nitrite giving NO and ascorbate radical that can be recycled back by mechanisms that I'm not going to discuss here. Okay, so this redox cycle can occur. Then what we have done, I'm going to skip these slides too. What we have done is to use several models to show that giving in the diet to these models nitrate, we could observe. So this is one model, the permanent bilateral occlusion carotid models. So in all these models, in this hyperperfusion model, and also in the type 2 diabetic model, we have observed that nitrate supplementation rescues cognitive function via improving hemodynamic and maintaining the bioavailability of nitric oxide. So this is an observation made in several models and the diet, okay, and with different stimuli, not only with glutamate, but also with whisker stimulation. So these preclinical models demonstrate the feasibility of developing a diet-dependent strategy to improve neurovascular coupling in individuals at risk for vascular cognitive impairment. And that's what we are now doing with these clinical trials. So this is the final slide to conclude that under conditions, hypoxic conditions or mild hyperperfusion, where this enzyme is not operative, there is a rescue mechanism relying on redox interaction of ascorbate and nitrite in the brain. And this redox interaction is functionally linked to neuronal activity because the ascorbate is coming from neurons upon glutamate stimulation. So the beauty of this rescue mechanism, maintaining the bioavailability of NO to maintaining the functional connection between neurons and blood flow, is that is modulated by diet. Okay, so I think that we have supported a mechanism that might not only prevent, but according to the preclinical models, enhance cognition. So we are not here preventing the cognitive decline. We are here enhancing cognition. In these preclinical models, we see that the animals learn faster, it preserves memory. So this is cognitive enhancement and not the prevention of cognitive decline. And so this is the mechanism that I've already described. So let me just thank all my group, particularly Rui Barbosa here, Kátia Lourenço, João Gonçalves, Ana Ledo, and also our collaborators in the Magnetic Resonance Imaging Center, Miguel Castelo Branco, our collaborator in Neurology Department, and also to our dear friends and long-time international collaborators, Enrique Cadenas in Los Angeles, Rafael Radi in Montevideo, Uruguay, Jon Landberg in Sweden, in Karolinska, and Greg Gerard in Lexington. And thank you all for your attention. And I'm sorry I went a little bit further than 30 minutes. Thank you very much. You are mute, Alexandre. Alexandre, you are mute.
Absolutely, my bad. So thank you for this very compelling and beautiful talk. There was already a couple of questions from the chat, but we will leave them for the end of the three talks, as I mentioned in the beginning. So next in line is Professor Mutai Aslan. She's currently a laboratory supervisor at the Agdeniz University Hospital, where she's also a lecturer at the Faculty of Medicine. And she's recently been elected to the Advanced Course Committee of the FEBS Committee. Professor Aslan has published so far more than 100 publications in peer-reviewed journals. plus patents, and she has currently an H index of 35, so very authoritative speaker. And today she's going to talk about oxidative stress and hemolysis, dependent destruction of vascular function in sickle cell disease. Please, Musai, the floor is yours.
Yes, thank you for that nice introduction. And I would also like to thank the organizers for giving me the opportunity to present my work. And today I will be talking about how oxidative stress and hemolysis destruct vascular function in sickle cell disease. So my slides are not moving. I don't know why. I cannot change my slides. Okay, there we go.
Is it working? Okay, perfect.
It's working right now. It's well known that sickle hemoglobin results from a point mutation in the gene encoding the beta chain of hemoglobin. And as a result of that mutation, the sixth amino acid, which is glutamate, changes into valine. And glutamate, as we most of us know, is a polar amino acid, and that gets replaced by a hydrophobic amino acid. And when the red cell is deoxygenated, that valine residue fits into a hydrophobic pocket in the near side beta chain. And when the deoxygenation is prolonged, then that polymerization leads to the change in the shape of erythrocytes from normal to sickle cell. Now, it is known that the time required for sickle hemoglobin polymerization prevents most erythrocytes from sickling in vivo. What is meant here is that for the change, for the hemoglobin polymerization to be prominent and to lead to the change in the sickle cell formation, erythrocytes require a capillary transit time to be more than 20 seconds. However, the capillary transit time of erythrocytes is nearly 17 minutes, and the time spent in the deoxygenated time is near 16 to 17 seconds. So what really occurs is when the capillary transit time exceeds 20 seconds, then we can see the sickling of the erythrocytes and vascular occlusion that is very typical of sickle cell disease. And this leads to pain, acute chest syndrome, pulmonary hypertension, and stroke. So, can we change the slides here? Okay. What has been shown is that there is increased red cell adhesion in sickle cell disease, and this leads to the increase in capillary transit time of erythrocytes. So it has been shown that there's an increase in Lutheran glycoprotein in the erythrocytes that binds to laminin. There's an increase in the alpha-4 beta-1 integrin that binds to VCAM1 in the endothelial cells. There's also an increase in Lutheran in the glycoproteins in CD36 and CD47, which bind to thrombospondin in the plasma. And there's also increase in CD41 and CD61 that bind to Wolff-Willer factor. So this increase in adhesion molecules in the red cells lead to a delay in the capillary transit time of erythrocytes in sickle cell disease. Now, okay, let's move the slide. Can we move the slide? Okay. There's also increased leukocyte adhesion in sickle cell disease, and studies have shown that MAC1, VLA4, P-selecting glycoprotein ligand are all increased adhesion molecules in the in the leukocytes, which bind to endothelial ligands that are also found to be increased in sickle cell disease, like MAC1 binds to ICAM1, 2, 3, but BLA4 binds to VCAM1, and P-selectin glycoprotein ligand binds to P-selectin and E-selectin. So this increase in adhesion molecules seen in sickle cell disease results in delayed capillary transit time. So vascular pathophysiology of sickle cell disease is can be summarized so far as adhesiveness of red and white blood cells to the endothelium, increased coagulation, and hemostatic perturbations. But the main topic of my talk today will be the nitric oxide dependent impairment of vascular function in sickle cell disease. So there is increased nitric oxide consumption, and as a result, there's impaired vascular function in sickle cell disease. Yoav very nicely summarized nitric oxide oxygen radical interactions. So we know that there's a potent influence on pathways of tissue oxidative and nitrosative chemistry when superoxide reacts with nitric oxide, but nitric oxide can react with catalase and cause inactivation of catalase. Nitric oxide can react with iron centers, and it can also react with lipid radicals. These reactions all lead to increased consumption of nitric oxide. I started working with sickle cell disease with my PhD thesis, and in my work initially we used a mouse model of sickle cell disease, and this is a double knockout model where there is breeding of 3 different mice to produce a mice that exclusively synthesizes the human sickle hemoglobin. Mice beta and alpha hemoglobin chains have anti-sickling properties. So to create a mouse model that completely mimics human sickle cell disease, they need to be knocked out. And the mice that exclusively synthesizes the hemoglobin chains in the human and the beta and the sickle cell hemoglobin is created by using these mice model. So these mice completely mimicked the symptoms that we see in human sickle cell disease patients. Okay, So we started out with these mice, and we looked at increased sources of reactive species production in these mice. We first measured human plasma xanthine oxidase activity and sickle cell disease mice plasma xanthine oxidase activity, and we saw that there was a significant increase in both the activity and the protein of xanthine oxidase in the circulation of these sickle cell disease mice. Again, what was striking that when we looked at the liver, we saw that there was a decrease of xanthine oxidase in the liver. And there was also increased ALT, which shows liver destruction occurring. And we also found increased aortic xanthine oxidase activity in the mice. So our initial hypothesis was that the hypoxia reoxygenation occurring in these sickle cell disease mouse led to and xanthine oxidase is located near the central vein regions in the liver, which are very susceptible to hypoxia reoxygenation. So as a result of this damage, the liver was releasing xanthine oxidase into the circulation, and this was resulting in the binding of xanthine oxidase to the vascular endothelium and an increased production of reactive oxygen species. And in fact, when we looked at superoxide production from these control mice and sickle cell disease mice, we could see that there was increased superoxide production, both basal and when you incubate the vessels with xanthine, you were seeing increased superoxide production. When we used xanthine oxidase inhibitor, allopurinol, or SOD, or SOD mimic manganese porphyrin, or a SOD inhibitor, we could see that there was a significant decrease of vessel superoxide production. And as a positive control, when we incubated the cells with BMAQ, we could see an increase in the reactive oxygen species production by the vessels. So this is looking at vascular relaxation in sickle cell disease mice. And we could see impaired acetylcholine-dependent vascular relaxation in sickle cell disease mice, and when they were also treated with L-arginine. But this is looking at the response from control vessels, and this is the response we get from sickle cell disease vessels. When we incubate the vessels with a manganese porphyrin compound, an SOD mimetic, we could see that there was partial restoration of acetylcholine-dependent vascular relaxation. And when we incubate the vessels with sodium nitroprusside, we could see that both nice relaxation response in both control and sickle cell disease vessels. So let's move on if I can. I cannot move my slides here.
Okay.
So this is looking at the binding of xanthine. This is confocal imaging, where we can see endothelial xanthine oxidase binding and transcytosis. This is the basal side, this is the apical side, and there's binding of xanthine oxidase on the endothelium, which gets transcytosed. So we knew now that there was increased release of xanthine oxidase from the liver into the circulation, which bound to the vascular endothelium, which led to increased superoxide production, that led to increased nitric oxide consumption and destruction of vascular function was a result. So I cannot move my slides. I don't know why. Okay.
The keyboard is not working.
Are you sure? It was working when I tested it. Well, the vascular nitric oxide interactions add molecular insights into oxygen-dependent tissue injury processes, as discussed. So the formation of peroxynitrite leads to generation of secondary species, leading to oxidation, nitration, and nitralization reactions. It also leads to vascular smooth muscle cell proliferation. It leads to cytoskeletal and matrix protein modifications. So moving on, we wanted to see we wanted to analyze liver and kidney proteins for protein nitration. So this is a 2D gel of liver and kidney.
Musa, sorry, are you changing the slides? Because I only see the old immunofluorescence for the xanthine oxidase.
Yes, I have changed the slide. I have.
So maybe it's not
now
showing.
I'm seeing the 2D gel fragments. Yes. No, it's moving on. So
okay,
now
the
slides are changing, and I'm not changing them. So I don't know.
Okay, Marco.
You may try and stop this the share and
maybe
share again.
So try to stop the sharing and share it again, Mutai, please.
Okay, sure.
Maybe we're stuck into.
So shall I share again?
Yeah,
you can try share again.
Okay, so let's see if it's moving.
Try to move along with the slides.
Back and forth.
Change.
With the mouse as well.
Okay, now it's working.
Now it is. Okay.
Perfect.
So
thank you.
Thank you. So we want to analyze sickle cell disease liver and kidney tissue homogenates for protein nitration, and these are 2D gels stained with Comasti and anti-nitrotyrosine antibody. We could see these blots here indicative of nitrated proteins. So we wanted to analyze and see what they were. And Molotov MS analysis showed us that those proteins were actin. And we also wanted to see which tryptic fragments were nitrated and we could see this typical mass spec pattern here, showing us nitration of tyrosine residues 91, 198, and 240. And these are the tryptic fragments which they correspond to. So we wanted to see how this nitration could affect actin function. And this is looking at the actin monomer, and this is where the ATP binding site of the actin monomer. This is the protein binding site of the actin monomer to myosin. As we all know, the actin monomers bind, one monomer binds to the other, and we get formation of fibrous actin. And these are the contact sites where one monomer, where one actin monomer contacts the other one. So this is contact site 3, and this is contact site 4. So we wanted to see the tyrosines that were nitrated on this actin monomer and what they were, and where they were structurally. So this is nitration of tyrosine residue 198. This is 240, and this is 91. So this is a space-filled model where you have the polymerization of actin. And this is when you have the polymerization of nitrated actin. So we wanted to look at the polymerization profile of nitrated actin at residues 91, 198, and 240. And what we saw was when actin was nitrated, there was an initial speed up in the polymerization, which we followed with the increase in fluorescence. And then there was a sudden, there was a drop in the polymerization. So there was, it was initially polymerizing, but then the effect of nitration led to the decrease of this polymerization. This is looking at the kinetics of polymerization. We can see that nitrated actin, although there's an increase initial polymerization, it falls off, and there's a decrease in the rate of actin polymerization when actin is nitrated. So this is looking at actin staining in control and sickle cell disease human tissue. Here you can see that actin is clogged and aggregated in the kidney sections from human sickle cell disease patients. So moving on, we also know that vascular inflammation in sickle cell disease is important, and there is increased interleukin-6 levels, interleukin-1, TNF-alpha, interleukin-8 levels in the serum, in the circulation of sickle cell disease patients. We know that tissue macrophages secrete interleukin-8, and the tunica media in the vessels also produce interleukin-6, and these can lead to the activation of polymorphonuclear leukocytes and the degranulation, leading to increased lactoferrin and elastase levels in the circulation of sickle cell disease patients. And there's also increased interleukin-1 and TNF-alpha production by monocytes in sickle cell disease patients. And these all lead, these also aggravate the adherence of these activated neutrophils to the endothelium. The increase in these cytokines lead to the increased synthesis of acute phase proteins in the liver. And these are alpha-1 antitrypsin, ceruloplasmin, alpha-2 microglobulin, and C-reactive proteins. So knowing that there's vascular inflammation in sickle cell disease, we wanted to look at the modulation of redox pathways in neutrophils from sickle cell disease patients. So what we did is we isolated neutrophils from control patients and from sickle cell disease patients. We looked at the superoxide released from these neutrophils. We looked at the peroxidase activity and the GSH content in these neutrophils, we saw that under basal conditions, there was not a significant difference between superoxide released from control and sickle cell neutrophils. When they were activated by FMLP, we could see this increase in superoxide release, which was not significantly different between control and sickle cells. And we couldn't see a significant difference in peroxidase activity, but there was a decreased amount of GSH in sickle cell neutrophils. And looking at inflammatory proteins, we saw that there was no significant difference between COX protein, but there was an increase in COX-2 protein in sickle cell disease neutrophils. And this is looking at the total COX activity. And this increase is mainly due to the increase of COX-2, which influences inflammation that we do observe in sickle cell disease patients. There's also increased protein of increased INOS here. These are the control and sickle neutrophil homogenates. We could see that the protein was increased nearly twofold in sickle cell disease neutrophils, and there was also an increase in nitrite levels that we did measure in sickle neutrophils. So let's move on, hopefully we can. So the redox, so the central aspect, like I said, of sickle cell vasculopathy is the impairment of endothelial regulation of vascular vasomotor tone, thrombosis, and inflammation, and the observed decrease in GSH levels in the neutrophils and increase in COX-2 and NOS2 protein content in the circulating neutrophils in sickle cell disease patients can aggravate inflammatory conditions associated with the disease. Now, as we know, polyunsaturated fatty acids are important in inflammation because the both inflammatory and anti-inflammatory eicosanoids are synthesized from different polyunsaturated fatty acids on this pathway. So there's the omega-6 polyunsaturated fatty acid pathway that starts with linoleic acid. It moves on to form arachidonic acid, and with COX and LOX activities, you have the formation of inflammatory eicosanoids here. And the omega-3 polyunsaturated fatty acids start with alpha-linolenic acid. They form eicosapentaenoic acid, and the effect of COX and LOX here forms eicosanoids that have anti-inflammatory properties. And this pathway leads to the formation of docosahexaenoic acid. And with the activity of LOX, you can also have formation of different anti-inflammatory eicosanoids by this pathway. So basically, the ratio of the omega-6, omega-3 polyunsaturated fatty acids determine the oxidative milieu in the tissue and in the circulation. So we wanted to analyze plasma polyunsaturated fatty acids in patients with homozygous disease. So we measured the homogamma-linoleic acid, arachidonic acid, eicosapentaenoic acid, and docosahexaenoic acid in 23 sickle cell disease patients and in controls. And what we observed was there was an increased omega-6 production, omega-6 polyunsaturated fatty acids, the homogamma-linoleic acid and arachidonic acid in sickle cell disease patients. And there was a significant decrease in the eicosapentaenoic acid and docosahexaenoic acid, which are omega-3 fatty acids in sickle cell disease patients. So when we look at the omega-6, omega-3 ratio, we can see a significant increase in these sickle cell disease patient plasma, which will also contribute to the inflammatory milieu present in sickle cell disease patients. So intravascular hemolysis and decompartmentalization of hemoglobin is also very important in increased consumption of nitric oxide. So if there's hemolysis, you get the release of heme, a free heme, and that iron-bound hemoglobin can react with nitric oxide in the vasculature and lead to increased nitric oxide consumption. So what we wanted to see was look at the inflammatory, the hemolysis markers in patients, in sickle cell disease patients and in controls. So in this study, we obtained blood from 25 homozygote sickle cell disease patients and 15 controls. Here you can see the variant hemoglobin analysis in these sickle cell disease patients. As we expect, there's increased in hemoglobin F percentage and hemoglobin A2 percentage. We cannot see normal hemoglobin in homozygote sickle cell disease patients, but instead we see hemoglobin S. So in these patients, we looked at laboratory values of that would be indicative of hemolysis. And under hemolysis, there's decreased hemoglobin concentration, which we saw was significantly lower in sickle cell disease patients. The percentage of hematocrit was significantly, again, decreased in sickle cell disease patients. Their increased reticulocyte count is a very nice indicator of hemolysis, which we were seeing was significantly increased in these sickle cell disease patients. There was increased midcorpuscular volume and midcorpuscular hemoglobin. Another indicator of hemolysis is total bilirubin and direct bilirubin levels, which were significantly increased in sickle cell disease patients. There was the total LDH activity was significantly increased in sickle cell disease patients. So we wanted to perform lactate dehydrogenase isoenzyme electrophoresis to know which fraction of LDH was increased. And we saw that the LDH1 fraction here, as we can see, was significantly increased, and LDH1 is mainly found in erythrocytes. So this increase of the LDH we were seeing in sickle cell disease patients resulted from increased LDH1 released from hemolyzed erythrocytes in sickle cell disease patients. There's also a decrease in haptoglobulin levels, which is also a nice indicator of hemolysis, because when cell-free hemoglobin is in the circulation, it binds to haptoglobulin that is synthesized by the liver, and it returns back to the liver to be degraded, to protect against oxidative injury in the vasculature. There was increased leukocytes, which shows the presence of inflammation, and also increased platelet count in sickle cell disease patients. So in this study, we could understand that hemolysis was also a significant contributor to increased vascular, we could also understand that hemolysis could be an underlying reason for increased nitric oxide consumption in sickle cell disease. So let's move on. Okay, if I can. Here, we know that altered lipid content in red cell membranes can lead to increased susceptibility to hemolysis. So the erythrocyte outer leaflet is mainly composed of phosphatidylcholine, sphingomyelin, and cholesterol. So a probable decrease in sphingomyelin cholesterol content in sickle cell disease could lead to increased susceptibility to hemolysis. So in this study, we analyzed serum lipid concentrations and LDL and HDL subfractions in sickle cell disease subjects. So in the 45 homozygote sickle cell disease subjects that were analyzed in this study, we could see a significant decrease of total cholesterol levels, LDL cholesterol and HDL cholesterol. On the other hand, there was increased triglyceride levels, which reflected itself with increased VLDL cholesterol. This is looking at the LDL subfraction analysis in sickle cell disease patients and in controls. We can see here that all the midbands and the LDL subfractions were decreased in sickle cell disease patients, especially the midbands CBA and LDL1 subfraction was significantly decreased in sickle cell disease subjects. This is looking at the analysis of HDL subfractions. Here are the gel scans of the control and sickle, what we see is that there, again, in all the subfractions of LDL, of HDL, the large, the intermediate, the small, we can see a significant decrease in the HDL subfractions in sickle cell disease subjects. So we know that there's also a decreased circulating cholesterol and LDL and HDL cholesterol and lipoproteins, in fact, in patients with sickle cell disease. Now, like I said, sphingomyelin is also important in membrane structure. So we wanted to analyze sphingomyelin and ceramide content in sickle cell disease patients and control. We know that de novo sphingomyelin synthesis can occur in the endoplasmic reticulum. It starts with the condensation of serum and palmitoyl CoA to form dihydrosphingosine, dihydroceramide. And when there's a phosphocholine added to the ceramide, we have the formation of sphingomyelin. So we have set up a multiple reaction monitoring method to analyze sphingomyelin and ceramides by LC-MS. And basically, in the first quadrupole, we do a single ion monitoring where we scan for the intact lipid. And then in the Q2 quadrupole, we fragment the lipid of our interest. And then in the third quadrupole, we again do a single ion monitoring and monitor the product ions. Now with sphingomyelins, there's a loss of this phosphocholine, which has a mass to charge ratio, detected mass to charge ratio 184. And when analyzing ceramides, we have the release of this fatty acid, and we have the product ion of sphingosine, which has a detectable mass to charge ratio of 264. So this is looking at the positive ion spectrum of these sphingolipids, 16, 18, and 24 carbon sphingomyelin. As the length of fatty acid side chain increases, you have an increase in hydrophobicity, so you detect them later in your scan. And this is looking at ceramides, 16 to 24. Again, the analysis time increases when the side chain of the fatty acid increases. So using this method, we could see that this 18 and 24 carbon chain containing fatty acids containing sphingomyelins were significantly decreased in the circulation of sickle cell disease patients. And again, the long chain ceramides were decreased in sickle cell disease patients. So in summary, what I'd like to say is that the participation of nitric oxide in oxidative reactions causes a reduction of nitric oxide bioavailability, which does contribute to vascular dysfunction in sickle cell disease. Now, markers of hemolysis are significantly increased in sickle cell disease patients, and the hemolytic rate has been shown by other investigators to be closely correlated with nitric oxide consumption. Now, the reduced cholesterol and decreased sphingomyelin content in sickle cell disease can certainly influence increased susceptibility to hemolysis, and therapeutic strategies that are designed to counteract endothelial inflammatory and oxidative abnormalities may reduce the frequency of hospitalization, blood transfusion, pain, the occurrence of acute chest syndrome, and pulmonary hypertension in patients with sickle cell disease. Now, I'd like to end my talk here by acknowledging my funding institutions in Turkey, the Scientific and Technological Research Council of Turkey and the Akdeniz University Research Foundation. I would also like to acknowledge my mentor, Bruce Freeman, whom I worked with at UAB, and I started my sickle cell disease studies with him. And Tim Towns and Thomas Ryan, I worked with. They also created the double knockout mouse of sickle cell disease. And Dale Parks, who has retired now, has helped me with the measurement of xanthine oxidase in plasma and in Western blot analysis. My lipid work started with my collaboration with Bülent Mütüz at the University of Windsor, Ontario, Canada. I would also like to acknowledge him. The clinicians I work here, the hematologists, Ozan Salim, Afan Küpesiz, very kindly provided me with the homozygous sickle cell disease patient plasma and blood. And my graduate students who work in my lab have contributed greatly to my work here. And I would like to thank everybody for listening. Thank you. I'm sorry for the delay in the slides, that technical problems.
Hello, sorry, my connection fell down, so I had to solve it another way. So Mutai, thank you for your very nice presentation. I was up till the end just a moment before the acknowledgement, so thank you very much. It was absolutely involving. The third talk from today, and the last one, of course, last but not least, is Professor Rita Maccarone from University of L'Aquila. Maccarone is a professor of physiology. A research field mostly focuses on functional study of the visual system and the mechanisms underlying retinal neurodegeneration. In particular, the scientific interest is focused on the degeneration of the new retina and retinal pigment epithelium, on the involvement of retinal vascular system, and on the breakdown of the hematoretinic barrier. And today she is going to talk about nanoserif particles as eligible candidate for counteracting oxidative stress and preserve retinal morphology and function in an animal model of age-related macular degeneration. So Rita, thanks. The floor is yours.
Okay. Good afternoon, everybody. Thank you very much, Alessandro, for the introduction, and thank also Mauro and the organization committee for the invitation and the opportunity to share with you my research. First of all, I would like to do a brief introduction on the visual system, the main topic of my studies. The eye is of the sensory system, is one of the sensory systems, and as you can see in this slide, is composed by many tissues, and all of them concur to our visual perception. We concentrated our research on the back side of the eye, where the retina and the pigmented epithelium are located. The retina is part of the central nervous system and is organized in layers that include many types of neuronal and glial cells, and they make synaptic connection with each other in a specific layer called the plexiform layer. The inner layer is composed by ganglion cells, and their axons form the optic nerve that by action potentials drive the information of the visual area of the central cortex. The external segments of photoreceptors, rods and cones, are closely connected with the pigmented epithelium, and their functions are dependent each other for the physiological visual cycle. In fact, as you can see in this cartoon, the molecules like 11-cis-retinal and all-trans-retinol used by photoreceptors to convert light in electrical signal are synthesized, released, and recycled by the pigmented epithelium. So, its connect functionality is fundamental for the visual perception. Furthermore, the pigmented epithelium concurs to recycle the external disk of photoreceptors, eliminating part of them by phagocytosis and autophagic mechanism. The pigmented epithelium has another important role, that is the synthesis and the release of the antioxidant molecules, like superoxide dismutase, catalase, and so on, with the aim to maintain the homeostasis in this microenvironment where the high metabolisms causes oxidative stress. In physiological conditions, the pigmented epithelium that, to simplify, I will call RPE from now, is part of the blood-retinal barrier together with the external component of the retinal vascular system called the choriocapillaries and with the Bruch membrane. The integrity of this barrier is a necessary condition for the maintenance of the retinal homeostasis. But as the age progresses and the endogenous antioxidant protection is lost, and also smoking, genetic predisposition, high fat diet are the main risk factors for the development of age-related macular degeneration, a multifactorial disorder that may cause blindness worldwide in the elderly. All these risk factors induce oxidative stress upregulation and the upregulation of the reactive oxygen species and dysfunction of the RPE. Furthermore, lipofuscin accumulation and the vascular endothelial growth factor upregulation too occur. The choriocapillaries and RPE, in fact, release vascular endothelial growth factor and neurogenesis occur. One of the main aspects of the AMD. All these events drive to blood-retinal barrier breakdown, inflammatory events, and retinal degeneration in age-related disorder. The AMD can be classified in two different forms, dry AMD and wet AMD. In many cases, the wet could be a progression of the dry form, but not necessarily. The patients, fundus oculi, show drusen accumulation starting from a specific region of the retina called macula, the area centralis with the main visual acuity. Instead, in the wet form, also hemorrhagic spots like these are visible. These phenomena induce a visual loss starting from the center of the visual field, and this is what the patients can see. That is a dark spot in the center of the visual field, more or less large, depending on the severity of the degeneration. Current clinical treatment for the dry form of AMD, where you can see the drusen, are based on multivitaminic diet supplementation, like beta-carotene, lutein, zeaxanthin, and so on. Instead, the gold standard treatment for the wet form of AMD, like this, consists in intravitreal injection of different drugs, all of them based on vascular endothelial growth factor antibodies like bevacizumab, aflibercept, and so on. And the main goal of this treatment is to block the effect of vascular endothelial growth factor upregulation on the neoangiogenesis event. All these therapies approaches are not resolving therapies, and in particular for the wet AMD, the limitation is due to that high frequency of intravitreal injections are needed to maintain the effective concentration of drugs in the eye. For the reason, this approach is too invasive and causes many side effects, such as, for example, the ocular inflammation. The goal of our study is to find an alternative and effective treatment by acting directly on oxidative stress and therefore blocking the degenerative events upstream of the cascade. To reach this goal, we performed in vivo experiments setting up an animal model of AMD based on the main risk factor to which we are all exposed, that is the continuous light, natural or artificial. Our model, in fact, consists to expose the albino rats on high-intensity light, 1000 lux, for 24 hours. As you can see in this photo, this picture. This model was well characterized in our laboratory, and some papers are published to describe the many features of AMD, and we demonstrated that this light damage model reproduces the main feature of both dry and wet AMD, depending on, as you can see, the recovery time after light damage, after light exposure. In fact, we demonstrated that 7 days of recovery drusen are accumulated in subretinal space between the RPE and photoreceptors, and after 60 days, a new vascularization occurs and new vessels invade the retina. I will show you the most relevant results that we obtained in our studies. This is a retinal cryosection where you can recognize the RPE labeled in green and all the nuclear layer in the of the retina in blue. By tunnel assay, we labeled the dying cells. And as you can see in the red dots, immediately after 24 hours of high light exposure, photoreceptor and epithelial cells start to die. I reported in the graph the high significant increase of dying cells in the RPE that happens 24 hours after photopic injury. In our model, the degeneration begins in a limited dorsal region that mimic the macula in the human AMD. In this hotspot, we found the autofluorescence lipofuscin granules that are called drusen in the human dry AMD, accumulated in the subretinal space. At the same time, as you can see down, the monolayer of epithelial cells loss its natural morphology, as you can see by the red row, and increase in size, characteristic changes of epithelial mesenchymal transition that occur in epithelial cells before dying. So, after photopic injury, the retinal barrier breaks down. To better understand the functional changes after photopic injury, we performed the electrophysiological recordings, and we compared the electrical response of the retinal cells before and after photopic injury. The ERG recordings were performed by increasing the intensity of flash stimulus, and here I reported the analysis of the two main waves that represent the response of photoreceptors, A wave, and the postsynaptic neuron, like bipolar cells located in the inner retinal network, the B wave. We follow the retinal function up to 120 days after light damage, as you can see here. The response significantly disappears compared to the control, both for A and B wave. Other relevant features of the wet AMD are demonstrated in our model. The vascular endothelial growth factor upregulation increases over time up to 120 days, as you can see by Western blot quantification. The vascular endothelial growth factor is purely detected in normal retina as well as immediately after light damage. The immunolocalization shows in red that this factor increase at 7 days photopic injury and invades all the retinal layer, as you can see here, starting from the RPE and the photoreceptor layer. Furthermore, we focused our attention on the outer nuclear layer to understand what happens after photopic injury about the microglia activation and the new vessel formation. We labeled the vessels in green and the microglia in red in retinal cryosections, as you can see in these confocal images, and in particular in the high magnification images. Both the vessels and the microglia invade the photoreceptor layers and a clear signal of degeneration occurs. We also quantified the percentage of area occupied by vessels and the number of the new tuft that represents the starting point for the growth of the new vessels, as is reported in the graph. And you can see that the number increases over time. So, after the characterization of our model, our aim was to test a nanotechnological approach to try to reverse the progression of the AMD in our animal model. Our interest of cerium oxide nanoparticles comes from the opportunity to take advantage of their peculiar characteristics that are both the nano dimension and the well-known antioxidant properties. Is well known demonstrated, in fact, by our collaborators at the University of L'Aquila, who synthesized and characterized the nanoseria particles, that they scavenge free radicals by switching their valence state from +3 fully reduced to +4 fully oxidized. And they can do this reaction thanks to the oxygen vacancies on their surface, and thanks also to their non-stoichiometric state. For these chemical properties, the nanoseria particles are classified as the pure antioxidants. This information obviously comes from my colleagues, Professor Maurizio Passacantando, that synthesized and characterized these nanoseria particles. So he knows very well the chemical and physical characteristics. So this is not my specific expertise, but obviously I trust in this statement. So the peculiar characteristic of this compound prompted us to test the possibility of a single application, in particular an intravitreal injection, to obtain prolonged efficacy over time and solve the limitation of intravitreal injection currently performed in the clinical treatment. For this reason, we set up 2 different experiments performing a single intravitreal injection in our animal model before and after photopic injury, and we followed the animal up to 120 days. Here I reported a schematic representation of this procedure. First of all, to demonstrate the effect of nanoseria particles on the oxidative stress, we investigated an endogenous molecule that play a pivotal role on the oxidative stress in the retina. And this is we demonstrated that the acrolein is upregulated after light damage, and that nanoseria particles, as you can see in the confocal images, significantly reduced the acrolein expression in the retina. The confocal images represent the high magnification of the photoreceptor layer, only the photoreceptor layer, and we detected a significant reduction when nanoseria particles are injected, demonstrating their antioxidant effects. The conjugation of nanoseria particles with fluorescein allow us to localize them in the tissue, and we found the nanoseria particles into the RPE cells after a single intravitreal injection, as you can see looking at the green dots in the cytoplasm of these cells. To identify the RPE cells in the transversal cryosection, we labeled the tissue with this red marker, RPE65, the specific marker of the epithelial cells. This evidence prompted us to investigate the effect of nanoseria on the epithelial cells, as described in this cartoon, to evaluate the capability to maintain the integrity of the retinal barrier, remembering that as I already showed you, the photopic injury induces an epithelial mesenchymal transition before the epithelial cell die. Actually, new experiments are in progress in our lab to better identify the intracellular localization of nanosyria particles in the RPE by transmission electron microscopy. The results of the effect of nanosyria particles on the RPE are very impressive, as you can see in these confocal images on the flat mount of the RPE labeled with RPE 65 in red and the phalloidin in green. As I told you, RPE 65 is a cytoplasmatic protein expressed by RPE cells in physiological conditions. Instead, the phalloidin is a good marker to identify the cell's profile. These confocal images clearly show the progression of the degeneration of the monolayer of RPE cells and the clear effect of nanosyria particles to preserve the retinal barrier. So while it is well known that the neurons undergo apoptotic death during degenerative processes, particular attention should be paid to another mechanism, the autophagy. In particular, a molecular biologist, Yoshinori Osumi, Nobel Prize in Physiology and Medicine, 2016, affirmed that autophagy is the fundamental mechanism for maintaining cellular homeostasis, and that autophagy dysregulation is involved in the pathogenesis of many disorders, including neurodegenerative diseases, infections, and cancer. So, since the homeostasis of the retina is a fundamental condition to maintain the healthy state, we studied this mechanism in correlation to retinal degeneration, and we demonstrated that light damage induces dysregulation of the two main markers of the autophagy, LC3B2 and P62. And that nanosyria particles, you can see in these results, revert this process. At the same time, the apoptosis is also reduced by intravitreal injection of nanosyria particles. Another important result is on the modulation of the microglia. It is well known that after injury, the inflammation occurs in the retina and the microglia activate and migrate from the internal to external retina, the photoreceptor layer. The microglia, in fact, in pathological condition, turn from the resting to the activated form and release many factors like vascular endothelial growth factor and fibroblast growth factor 2. In our model, we demonstrated that microglia activate and migrate to the retina, but nanosyria particles are able to inhibit this phenomenon. Another important result is about the ERG recordings. The ERG recordings, shows that a single intravitreal injection of nanosyria particles is able to maintain the retinal function, and this is evident looking at the analysis of the A and the B wave performed as I previously described. Two different series of experiments was performed. In fact, the intravitreal injection in these experiments was done before and after photopic injury. And we demonstrated that the pretreatment is more effective, in red, to maintain retinal function, as you can see in the red line. But also the post-injury injection, the gray line, even if partially, is able to preserve retinal function. This is a relevant results that give hope for the potential efficacy of the nanoseria particles when the AMD is already in progress. In conclusion, we can affirm that nanoseria particles are effective to counteract the main feature of AMD. In fact, after a single intravitreal injection, the nanoseria particles are internalized into retinal epithelial cells, maintain their efficacy after light damage, reduce the oxidative stress, as highlighted by acrolein quantification, reduce the microglia activation, retinal inflammation, and vascular endothelial growth factor upregulation, and new vascularization, maintain the integrity of the RPE and the blood retinal barrier, reduce the apoptosis, maintain the physiological levels of autophagy, preserve the retinal function. So we can affirm that the nanoseria particles acting as a pure antioxidant could be a winner strategy to counteract the AMD. The future perspectives are to translate the results of this preclinical studies to the clinical application, but obviously further effort will have to be made. There are some limitations to overcome. The first limitation, for example, is due to that the nanoseria particles are not water soluble, but they are in suspension. So this makes difficult to develop a drug for human patients with a standardized dose. The second limitation is about the method of administration. In fact, to date, is not yet been developed a topical route of the administration by drops, because the cornea is impermeable to many drugs, and many studies are focused on this challenge. And this is the reason why the actual treatment of AMD is based on intravitreal injections. In any case, we do not forget that the eye are a very efficient drainage system in the anterior side. So the idea to develop the eye drops must take into account that there is the possibility that the drug could not be reached the retina. So I would like to show you my collaboration, who share with me the interest on this topic. This is my group, my team at the University of L'Aquila, and all my colleagues. The Lucia Poggi at University of Trento, and Christina Walter at the University of Australian National University. My colleague Krzysztof Turleski at the Nenki Institute of Warsaw in Poland, Dr. Marco Feligioni, Fondazione Ebri Rita Levi Montalcini in Rome, Cristiano De Pitta at the University of Padova, and Mirko Pesce at the University of Chieti. And I would like to thank you very much for your attention.
Thank you, Rita, for the very nice presentation. We also have a guest which was lifted from the academic duties.
Hi, Mutai. Hi, João. Hi, Rita.
Oh.
Hello, Mauro.
Hi, Mauro.
Mauro, hello. It's so nice to see you.
So for me, the same, Mutai. I'm really glad that I could partly join your lectures. I was fascinated. Of course, I could not follow them full time as they absolutely deserve to, but I'm sure that the audience learned a lot from you, and from the chat, I see that they also appreciated the contents and were very curious about your work, the information you deliver. So thanks again for being with us, and I leave Alessandro in his role of chair of this session.
Okay.
Mauro, bye.
Ciao, Rita.
So I believe there are a few questions. We have time for a few questions from the chat. The first ones are actually for João. The first one was, what is the effect of the nitric oxide on mitochondria of neurons and cardiomyocytes, and how much of nitric oxide is too much, in your opinion?
Okay, the effects of nitric oxide on mitochondria of neurons. Of course, yeah, cytochrome C oxidase is a heme protein. And so the fastest reactions of nitric oxide in terms of rate constants are the reaction of nitric oxide with heme centers. So for sure, because cytochrome C oxidase is very abundant, for sure cytochrome C oxidase is a major target for NO. And it is very well known, or a long time ago, that okay, the reaction is not straightforward, but under physiological conditions, depending on oxygen, the content of cytochrome C oxidase, what is known for a long time, for a long time, more than 20 years or something, is that NO competes with oxygen for binding to cytochrome C oxidase. So this was formulated by Alberto Boveris a long time ago. So soon after the discovery of NO as a vasorelax, as an endothelial-derived relaxing factor, it was realized that NO could somehow regulate oxygen consumption and ATP production by this way. So for sure, but in this particular case, so for sure that upon glutamate stimulation and NO production, mitochondrial within the diffusional spread of NO, okay, can be a target for NO. There is also a reaction depending on the abundance of oxygen, and also, of course, Moncada, Salvador Moncada was one of the leaders in this area. There is the possibility of cytochrome C, the reaction of cytochrome C oxidase yielding nitrite from NO. Okay, but the main point here is the reversible binding. But what we have observed in these particular models, that in rats, is that the main mechanism for NO inactivation in brain upon glutamate stimulation is the reaction with oxyhemoglobin in vessels, yielding nitrate, because, you know, there's a lot of hemoglobin. I mean, in the erythrocyte is about 50 millimolar. So my view is that, actually we are planning a grant on this topic. My view is that upon glutamate stimulation, NO can regulate mitochondrial respiration, but at the same time, there is plenty of NO to diffuse to nearby arteries and evoking this vasodilation. So it's a very, very interesting question.
So I actually have myself a question. I was wondering, maybe I missed it, so I really hope you didn't say and I just missed it, about the role of nitric oxide synthase in the cognitive decline in general, since it seems to be so involved through the neurovascular coupling. Is there any investigation which ascertained if there are different genes or alleles which are of the NOS, which are associated as danger factors in the development of cognitive decline, which might be just dementia or
I don't know, Alex. You mean just genes related with the nitric oxide synthesis?
Yeah, isoforms or alleles which might be pathological in development of cognitive decline.
I don't know. Anyway, the target for NO in the vessels are soluble guanylate cyclase, and this enzyme, this protein, the soluble guanylate cyclase, doesn't care if NO, which isoform NO comes from, or either if NO comes from nitrite, as I mentioned, because it's so it's interesting, but I don't know if there is any genetic investigation on this connection between expression of isoforms of nitric oxide, of neuronal isoform, and the hemodynamic response.
May I ask a question, Yao?
Yeah, of course.
I was very impressed by your real-time data showing the increase of nitric oxide and then the increase in blood flow and oxygen. That's very nice. I wanted to, could you comment on how much of that nitric oxide is coming from NOS and how much of it is coming from eNOS in the cerebral circulation?
In one of the slides I've shown, if we inhibit the endothelial isoform, nothing happens.
Yes, I see that.
Okay, so in this process, apparently the isoform involved is the neuronal isoform, not the endothelial isoform. And when we measure quantitatively the NO, we can measure up to 50 nanomolar, 100 nanomolar of NO.
And you say that most of that is coming from neuronal loss in the...
Yeah, but please note that there are several spots of nitric oxide synthesis, neuronal isoform, that are activated at the same time, okay? So when I say that we are measuring 50, 100 nanomolar, this means that this is volume signaling, that are several sources contributing to this signal. With our sensors, we don't know the number of your door. We know the area code of your street, of your road. Okay, you see, sensors sense a volume, and the metaphor is this one. With these sensors, we don't know the number of the door. We know the area code. Okay, so it's important to keep in mind that there are limitations in the use of these sensors.
The quick question is there any evidence that cytochrome oxidase can be nitrosylated?
Complex 1, yes. In the same magnitude, order of magnitude, NO can both inhibit cytochrome C oxidase and also nitrosate complex 1 in mitochondria. Okay, but I think that there are also some evidence, because the nitrosation reaction has a rate constant that is much slower than the reversible binding to the heme. Also, NO itself, there is some controversial about this, okay, with Stuart Lipton and so on, but NO itself cannot direct nitrosate. It has to indirectly produce a nitrosating species, like N2O3, like NO+, okay? So it's not such a straightforward reaction.
But in that case, do you recall, can you say that complex 1 is inhibited by nitrosylation, or is not known or not?
If you have a black and white situation, if you have 100% of the complex 1 in 100% of mitochondria inhibit, I mean, yeah, but no, it depends on the yield of the because not only, as far as I know, this when we think about the sparing capacity of the respiratory chain is not only at the cytochrome C oxidase level. Maybe there are other levels, okay? Because we have a lot more cytochrome C oxidase than that would be necessary to perform the respiration.
Okay, thank you.
But the question here is always the quantitative aspect of these issues. Of course, we can find nitrosation, we can find nitration, we can find inhibition. I mean, so what? If you have 1 million molecules of cytochrome C oxidase and 100,000 are inhibited, so what?
Depends on the equilibrium, of course.
Sure. Yeah.
I have a very quick question myself for Mutai about the GSH, which you saw diminished on the granulocytes, if I'm not mistaken.
Yes.
Do you have any, knowledge? Is there any knowledge about the molecular mechanism by which the GSH is depleted in granulocytes during the sickle cell disease?
Well, that's a very nice question. In my work, we could not see increased free radical formation. We couldn't see increased superoxide production. We looked at peroxidase activity. We couldn't see any difference in peroxidase activity, but somewhat there seemed to be an inflammatory stimulus in the polymorphonuclear leukocytes. So I guess that maybe with time that could lead to a buildup of inflammation-related reactive oxygen species production, and we were seeing actually the previously depleted GSH through these reactions. But when we measured superoxide production in our isolated neutrophils, we couldn't see a major difference in control.
So we don't know if it's a biosynthetic problem or there is more disulfide.
I don't know as far as my work, but I think it's a buildup because these neutrophils are more exposed to inflammation, and during the sickle cell crises, we see that there's increased inflammation also.
And if I may add to that, do you have any evidence that the pentose phosphate shunt is somehow altered in your system so that you don't have enough NADPH?
Yes, that could certainly be a reason, of course.
So maybe some key enzymes of that pathway might parallel.
Exactly, they may be involved, right?
Okay, thanks.
And last question for Rita too. I was going to ask something about the correlation between the model and the resolution of inflammation, but as a matter of fact, we are working on that, so there would be that wouldn't make any sense. So I don't think we have final results. Very simple question. In your opinion, the role of EPA and DHA, most of the times they say they're both very important in the working of the photoreceptor. Which one among them is the most important? Because they seem to be very important for the transition of the photoreceptor, but they never investigate them. They always say that they have different roles, but to date, I don't think we have any, at least to my knowledge, any definitive answer to that.
Did you ask about the nanoseria particles?
Not necessarily about the nanoseria, about the EPA and the DHA. I mean,
Okay.
omega-3 fatty
The
acid.
correlation between the two tissues.
I mean,
The
which one of them, in your opinion, is most important in the working of the photoreceptor? Because I never get a conclusive answer to that when I ask it.
If I understand well your question, which is the main important
Yeah, the most important.
between the photoreceptors and the RPE.
No, between the eicosapentaenoic acid or the docosahexaenoic acid in the working of the photoreceptor. No problems.
Okay, so you are asking about the role in the photoreceptor of the eicosanoid acid. So
which one is for you the most important?
Yeah. So the photoreceptors are the eye. A big problem of the photoreceptors here is that they are an high necessity of oxygen. So all the mechanisms involved in the stress, oxidative stress, and all the pathways that are upregulated after this event are fundamental for the photoreceptor to try to protect themselves and for this high consumption of oxygen. So eicosanoid, in my opinion, I think that are very important in this process to try to protect the photoreceptor from because the photoreceptor has this limit. They use a lot of oxygen, and they can go from the hypoxia to hyperoxia continuously because they don't have the oxygen support from the choroid. Cannot be modulated because the choroid release a quantity of oxygen, but in different moments the photoreceptor need more or less of oxygen. So they are in this unbalanced condition. So I think that the eicosanoids are fundamental to maintain the homeostasis of this system.
Maybe, Rita, when we understand better if resolving E coming from E or resolving D coming from DHA, which one is more relevant, then we might know that one omega-3 is more relevant than the other. But apparently we don't know yet.
We don't know yet, yes.
Okay, thanks.
Okay, I think this wraps up for today. By the way, this was the last appointment for this year, so it takes the occasion to wish you a very Merry Christmas, a Happy New Year.
Here.
Thank you for being
Thank you,
here.
likewise.
Thank you.
Merry Christmas.
A real pleasure.
Merry Christmas
to you. It was
and thank you very much.
It was a pleasure. Thanks
Yes,
for all
thank
the
you
others who
again.
connected in the meantime.
Bye.
Okay, have a wonderful holiday.
Thank you.
Okay.
Bye.
All of
Thank
you.
you very much
Enjoy
again.
it.
Ciao.
And have a nice time
Have a
during
good
the
day.
festivities for everybody. Bye-bye.
Ciao.
Ciao.
Bye,
Thank you.
everybody. Ciao.
Thank you again.
Ciao.
Ciao. Persone veramente